In this 3-mark subjective question, they are directing us to understand the statement, the use of AV isotope of a nitrogen by Matthew Mesler and Frederick Starr demonstrated semi-conservative mode of DNA replication. With this specification, they are directing us to explain how did they arrive at this conclusion. For this illustration, we are given the semi-conservative mode of DNA replication as per the NCERT pattern. In this particular first illustration, we are representing the 15N DNA formation in the E. coli. When you are growing the E. coli only with the 15N nitrogen, and the DNA which you are segregating from the E. coli, when we are separating in the centrifuge tube, we are observing this DNA band sedimentation at the base, illustrating this DNA is at the maximum weight. When you are growing the E. coli for the second time in the 14N nutrition medium alone, we will be getting the 14N and 15N in the one DNA set, so that we are understanding the semi-conservative mode of DNA replication. The reconfirmation will be done when we are going for the second time in the 14N nutrition medium alone, not with the 15N nutrition medium. When we are growing at the time, we are finding the two type of DNA formation. One DNA is having only 14N, the other DNA is having 14N and 15N. And when we are segregating this DNA in the centrifuge tube, we are observing the DNA band sedimentation in the middle as well as at the top of the centrifuge tube. This is exactly illustrating the semi-conservative mode of DNA replication. In this way, by narrating each one of the step, we are securing the 3 mark to satisfy this optional question demand. In this 3 mark subjective question, they are directing us to specify the translation mechanism in which way it is happening in the prokaryotic ribosome. For this, we are illustrating the translation mechanism in the sequential order by specifying each one of the step in a sequential order. First, we are starting with the charging of the tRNA or amino acylation of the tRNA. For this illustration, we are getting half mark. Subunit of the ribosome binds to the mRNA exclusively at the 5' end of the mRNA. For this specification, we are getting half mark. The ribosome, when it is bind to the mRNA at the start codon, that is recognized only by the initial tRNA. In this way, the translation initiation is executed. For this specification, we are getting another half mark. In the elongation phase, the amino acid with the tRNA subsequently binds to the appropriate codon of the mRNA, forming a complementary base pair with the tRNA anticodon. For this specification, we are getting half mark. Ribosome moves from codon to codon along with the mRNA, and amino acid are added one by one in the two sides of the larger subunit joined by peptide formation. For this specification, we are getting half mark. And we need to narrate the termination. In this particular segment, we are specifying the releasing factor when it is bind to the stop codon, either UAA or UAG or UGA. The polypeptide is getting released from the ribosome. In this way, the translation is terminated. For this specification, we are getting another half mark. In this way, we are satisfying the overall 3 mark demand of the subjective question.